name is Irina Filitovic, and I would like to show you a demo of my final project called Case Study in Biology. The goal of the project is to build a classification network that would predict a type of biochemical interaction called mechanism of action in pharmacology between a chemical compound and breast cancer cells given fluorescence microscopy images of the cells treated with the compound. This information is important for drug discovery. Here's what the images look like. I used 1200 images annotated with true mechanism of action labels from a larger broad bioimage benchmark collection dataset. I am going to implement a multi-scale convolutional neural network architecture, or MCNN, similar to the one described in the article cited below. But instead of using Caffe framework, which authors did, I will use Keras API with TensorFlow backend, recording my results on TensorBoard. The authors designed this network to solve the task of cell images classification into any types of classes provided during training and demonstrated high classification performance of the approach on 8 datasets, including the one I'm using. One of the challenges with cellular images classification is that of both biological and technical reasons, classes generally evident at different image scales. The authors claim that their architecture seamlessly deals with this challenge, training just one network with parallel sequences of convolutional layers. It's achieved by configuring parallel branches of layers, each starting with a max pool layer with different pool sizes, followed by one or multiple convolution layers, after which the outputs are brought to the same size using max pool layers again. Then the outputs are merged and passed through more convolutional and dense layers to produce the final predictions. All my code is written from scratch and consists of 3 Jupyter notebooks. Download data notebook downloads the annotated part of the dataset and saves it as a pickle file, which needs approximately 1.2 gigabytes of disk space. The full dataset consists of 55 zip files, approximately 750 megabytes each. However, they also contain images that are not annotated. The code reads one archive at a time and extracts images that correspond to the labeled examples only. Then each image is reduced in size by 50% and converted to an 8-bit range scale. The code aggregates the samples in a NumPy array to store them in a pickle file later on. It took me about 2 hours to run this notebook on the EC2 instance I was using. Train model notebook trains the best model I was able to construct and saves it as an H5 file. It loads the saved image data from the pickle file, then displays 3 examples images of each class. The mechanism of action classification is the task of making prediction for an unseen compound. Therefore, all images corresponding to one compound should either be in the training set or in the testing set. Therefore, I pick 5 compounds that will be used for testing. They come from the 5 largest classes, but have the smallest number of images corresponding to them in their class. And then I split my data set into training and testing subsets accordingly. Here I convert the labels to one-hot encoded labels and then define the augmentation techniques that I'm going to use. And plot augmented variations of an image. Then I define the model's architecture and train it. The results of training are logged in TensorBoard using Keras callbacks, and the results are written in the subfolders of the logs folder. Please refer to my report's experiment, challenges, and results section to track how I changed all these parameters in the model's architecture. Some modifications I tried include using additional augmentation techniques, changing the optimization algorithm, experimenting with 1, 2, and 3 convolutional layers per parallel sequence, adding dropout layers, applying batch normalization, it's a regularization technique, using less pooling, and removing layers after the outputs of the parallel sequences are merged. The way the model is configured in the submitted version of the notebook corresponds to the best model I was able to obtain. It took me about 4 hours to train it. Predict label for compound notebook uses the saved model to predict labels for test compounds. It loads the samples and the model, prints names of testing compounds and training compounds, and presents predictions for 5 testing compounds. In reality, mechanism of action is predicted for an individual compound concentration pair, so the final labels are calculated in a way that prediction for all images corresponding to a single compound concentration pair are averaged, and the class is determined from that average. In addition to that, for each test compound, I plotted an image example for every concentration. I ran my Jupyter notebooks on an EC2 instance of the same AMI we used during week 5 of the course. Configuration and usage instruction from week 5 lab is enclosed with my final project submission. Initially, I implemented the original architecture described earlier. I tried to select data augmentation techniques and set hyperparameters as close as possible to the ones the authors used. However, it still was an approximation. I tried 12 different model variations, experimenting with various hyperparameter settings, layer configuration, and regularization techniques. As you may see on my TensorBoard training and smooth validation accuracy plots, I did not succeed in fighting overfitting, and my best runs validation accuracy stagnates at 30%. Although I was not able to achieve high classification performance, I implemented a very neat MCNN architecture and tried many interesting techniques in action and got comfortable with Keras API. A lesson I definitely learned is that overfitting is a serious challenge when training a CNN on a small dataset. The novel MCNN approach proposed by authors processes multiple scales of an image over parallel sequences of convolutional layers, each of which takes care of resizing the same input image to the necessary scale. This allows you to discover features from different image scales in one go, without training several networks. Tuning hyperparameters takes a really long time and it might not lead to producing a successful model. Some more things I would like to try if there was more time. I would try different normalization techniques of initial sample images. I'd try 7 parallel sequences of layers instead of 3. And I would apply the original architecture to other datasets, as well as experiment with other weight and bias initializers. Thanks for watching. Goodbye.